عاود بعد رجع دوزو نورمالمون خص دوز وحده تكنيك.
The activity of taste cells is transmitted by fibers of 3 cranial nerves to the solitary tract nucleus of the medulla. Then this information is transmitted to the thalamus and successively to the gustatory area of the cortex, where it gives rise to oral perception. From the solitary tract nucleus, the gustatory information goes also to the amygdala and hypothalamus, where the hunger center is located. And this information is the input that can modulate the hunger center. Taste sensitivity varies greatly among individuals, which could potentially influence food choices and therefore the nutritional status and health of the individual. So, the ability to taste bitter of urea compound as PROP is the best known example of taste variability, and it's considered to be a paradigm of the general taste perception. Based on the ability to taste PROP, individuals can be divided into PROP taster category. And PROP sensitivity is mediated by TRS2R38 receptor, and its variability is associated with the allelic diversity of the gene due to the single nucleotide polymorphism that give rise to the common haplotype, the PAD, the dominant taster variant, and ABI, the non-taster recessive one. Also, super taster have a greater density of uniform papillae on the anterior surface of the tongue, and it could partially explain the greater responsiveness of super taster to a range of stimuli. However, other reports show no association between PROP taster status and these morphological and physiological variables. So, the main aim of this work was to investigate the function of taste and its mechanism controlling the individual variability and health of the individual. Many studies have led to controversial results, often due to the highly subjective morphological and sensory assessment, leading to significant measurement errors. So I conducted a multidisciplinary research work which integrated molecular biology and psychophysical approaches with machine learning and deep learning methods, addressing both the fundamental aspects of sensory neuroscience and translational application in health and disease. So, given the implication that the genetic variants of the bitter receptor have in function, tasting and non-tasting function and disorder, the first issue of this, the first aim of this work was the issue of assessing the efficacy of machine learning classifiers to automatically and highly accurate classify TIS2R38 variants by exploiting psychophysical and biological features which were used as predictive variables of the data set. So, to this aim, we used supervised learning classifiers to learn from a structured data set as sensory, genetic, morphological, demographic, and clinical features recorded by each subject, to evaluate the differences among participants and return with high prediction TIS2R38 genotype of novel sample. So we tested these 6 algorithms. An essential phase in the running of a supervised learning project is the data processing, which includes firstly the analysis of correlation between the chosen targets and features, and this step allows us to understand the relationship between variables and to verify the consistency of the data set. After that, a data cleaning phase is performed by handling missing values, outliers values, and eliminating the duplicates one. Then the data is prepared for the learning algorithm through encoding the categorical features and the normalization of the numerical one. Finally, a data balancing strategy is applied, addressing class imbalance to ensure that the model doesn't learn preferentially from the majority class. So after processing the data, we use these metrics to evaluate the performance of our models, and the cut booster classifier was the best performance one. The raw curve was another metric of evaluation which confirmed that the cut booster classifier best identify different genotypes with high precision. In fact, it made 96%, 78%, and 82% correct prediction of ABI, ABI, PAB, ABI, and PAB, PAB genotypes respectively. Sharp algorithm allowed us to obtain an overview of the most important features and for the model and how they are impacting it to make a prediction. A high feature value showed in red color positively contribute to the target class, while low feature value showed in blue color negatively contribute to the target class. And the analysis showed that when prop solution is low, the model makes ABI, ABI prediction. While when the value of this feature is medium or high, the model pushes the prediction towards the other genotypes. And this is an agreement with physiological data which indicated that the presence of two PAB haplotype as opposed to one doesn't add benefits to proper detection. So our findings suggest that supervised learning may represent a trustworthy and objective tool for identifying variants of these beta receptor, which by reducing cost and time of molecular biology could find wide application in text physiology and medicine studies. At this point, the question is, can be convolutional neural network a reliable automated tool to assess papillae density and distribution, offering new opportunities and objective large-scale analysis of fungiform papillae with potential application in both clinical diagnostic? To this aim, and also to overcome the challenges and limitations associated with the manual counting of fungiform papillae, we assessed 3 convolutional neural network aggregation tasks on 175 images of the tongue. The classic UNet, the multi-res UNet, and the optimized UNet that we specifically designed to enhance the performance of the model, also when it must identify fungiform papillae with challenging morphology. So here we have the workflow of how the data were processed, and it starts by the initial image of the tongue, followed by an ellipse detector which detects a particular region of interest, which is then cropped, standardized and normalized by the contrast-limited adaptive histogram equalization to enhance papillae visibility. The processed images and the ground truth images are then fed into the end-to-end deep learning models, generating output images featuring bright spots at the center of each fungiform papillae. So after processing the data, we analyzed how the model learned by using 2 metric errors as a loss function, and the average of error between the ground truth and the predicted values, and also the structural similarity and the dice coefficient between the ground truth and the predicted values. Both here, mean squared error and the mean absolute error decrease smoothly for training and validation sets, and the curves are strictly close to each other. And this result showed that the optimized method have the most stable and consistent learning behavior. Also, we found the lowest error and the highest similarity between the ground truth and the predicted values. In addition, by local peak detection analysis that extract the coordinate of spot of each fungiform papillae, we calculate the rate of true positive and true negative with respect to the ground truth for all predicted fungiform papillae by each model. Here, 3 representative examples of the ability of each model to identify fungiform papillae in challenging input images with different papillae density and morphologies, are shown in this figure. And from the point with different color, which indicated true positive and true positive and true negative, it's here. And from the table with that showed the predicted values, the optimized unit showed the most balanced prediction across all examples. So these results showed that the optimized unit achieved the highest stability, accuracy, and robustness in the learning and identification of fungiform papillae with challenging morphologies. And the ability to automatically detect fungiform papillae has important impact for understanding taste variability for individuals, which could eventually help in diagnosing taste disorder or maybe giving personalized nutrition plans. So at this point, we wanted to apply supervised learning regression method to analyze the overall taste status with high precision of healthy control and patient with chemosensory loss, and to establish which combination of parameters best predict the taste function in both groups. Also, since the oral sensitivity that we experience when tasting food consists of a multimodal interaction of taste, smell, and trigeminal, we also assess the supervised learning method to understand and to determine the significance and the contribution of the single component of gustatory, olfactory, and trigeminal. So to this aim, we used these algorithms to predict a continuous value output based on sensory, clinical, and anthropometric features previously recorded by each subject to return the value of the target. And from the scatter plot showing the experimental values versus predicted values obtained by the random forest regressor, and from the metric of evaluation, the random forest regressor was the best algorithm to predict the overall taste status with high precision in both groups. Also, we found a very low value of mean squared error, which represents the standard deviation of the distances from the regression line, and the low value of the mean absolute percentage error in both groups. The interpretation of the analysis of the random forest regressor, which link feature importance with feature effect by CHAP algorithm, allowed us to obtain the importance of feature for the model to make the prediction. And the analysis showed that salty low concentration and sour high concentration specifically characterized healthy control, while bitter high concentration and astringent high concentration mostly identified patients with chemosensory loss. So, in addition, we, to determine with high precision the significance and the contribution to the oral sensitivity of a single component of gustatory, olfactory, and trigeminal, we also built a supervised learning regression method trained on previously determined features of participants and returned with high precision the prediction of the oral sensitivity. And here, from the metric of evaluation, the cat-boost regressor this time predict values that were strictly close to experimental values. In addition, by the permutation analysis, explainer analysis, we obtain the importance and the contribution of the single component to predict the oral sensitivity by the cat-boost classifier at both levels. At the local interpretability level, meaning the analysis on the single instance, and on the global interpretability level, meaning the analysis on the whole data set. So in control individuals, we can see that sensitivity for astringency, spiciness, taste, and smell were the most important features with a positive contribution. While sensitive in patient individual, sensitivity for astringency, spiciness, and the PTC rating were the most important features with a negative contribution. And it is interesting to note that the global interpretability showed that in controls, sensitivity for astringency and spiciness had a similar impact, while in patients, sensitivity for astringency had a greater explanatory power, and the sensitivity for spiciness had a lower impact in patients. Also, sensitivity for taste in patients had a lower impact, and in control had more impact. So this result showed that supervised learning regression method is a reliable and highly precise approach to analyze the oral sensitivity in healthy control and patient with chemosensory loss, and to identify which stimuli alone or in combination best predict the overall taste status in both groups. In addition, the trigeminal component that we evaluated by astringency and spiciness were the most important in both groups. However, astringency and spiciness were the most important and gave a similar contribution in controls, while in patients, astringency was the most important feature. Finally, we used supervised learning algorithms to clarify the diagnostic significance of taste deficit by searching for data that correlate with specific features to automatically distinguish between patient with Parkinson's disease versus healthy control, and to make a prediction of the disease severity according to the Horn and Dior scale, classifying them into who had a high severity and those who had a moderate severity. So we used the algorithm that learned the relationship between features and the target to predict the correct class of new samples. And from the metric of evaluation, the cat-boost classifier was the best performance model to classify healthy control versus patient with Parkinson's disease. The interpretation of the analysis of the model to predict patient or healthy control showed that by a CHAP algorithm, which highlight the feature importance with feature effect, showed that the perceived intensity of NACL was the most important feature. And the description of a different quality of astringency was the most important features to predict patient with Parkinson's disease. Also, NACL and the threshold was the most important to classify healthy control, but here with a negative contribution. The NACL correct identification was the third important feature with a positive contribution. And these results mean that the model makes a prediction as a patient with Parkinson's disease if he or she perceived no taste for salty or described a different quality for astringency. And it makes or classify a healthy control if he or she identified correctly salty stimuli. This result of the classification of the disease severity of patient with Parkinson's disease by cat-boost classifier showed that longer disease duration and the akinetic rigid type predict high severity. And that reflects the natural progress of Parkinson's disease and the fact that this subtype is known to be more progressive, more disabling, and progress more rapidly. I point out that in this experiment we also used the polymorphisms of alpha-synuclein gene. And among the genetic features, CC genotype of this SNP was associated to predict the moderate severity, confirming its potential protective role, while TT genotype of this SNP and lower taste acuity predict high severity, strengthening the link with sensory decline and disease progression. Also, we found sex-based differences in the disease progression. This result showed that the cut-boost algorithm was the best to classify healthy control versus patient with Parkinson's disease, clarifying that these deficits are modality specific to with saltiness and astringency most affected. And the interpretation of results to classify patients based on the disease severity showed distinct pattern of features importance and directionality for each severity class, reflecting the complex interplay between genetic, sensory, and clinical features in the disease, in Parkinson's disease progression. So, in conclusion, these results suggest that when combined with supervised learning and genetic profiling, the sensory marker could offer a powerful non-invasive tool for the Parkinson's disease diagnostic and the disease stratification. Thank you for your attention.
Thank you everybody for this presentation. Allah.
Splendid.
Indication for clinical practice. Do you think your results could have indication in clinical practice in the next few years?
Yeah, absolutely yes, because the model we used was tested on our main cohort, but it can be applied on different hospital too, and maybe it can be a part also of clinical diagnostic.
Okay, thank you very much.
I was wondering, do you believe that this taste impairment in the future could be also considered among the